workflow I prepared, and I'm going to present a particular lecture on discussion of the physiology and mechanics of muscle contraction. As you know, and we've spoken throughout the entire semester, there's an interaction between body systems. It's important for us to understand that there's a homeostatic balance within all the body systems. Today I discussed nervous system and the muscle system in regards to how the muscular contraction will occur. There's an electrical stimulation through the nervous system down an axon to a synaptic end bulb. And what I'd like to display is simply this interaction between nervous and muscle system in order for us to understand the entire concept of how a muscle contracts. It's important to know the physiology. I'll discuss that in the end. I'll also discuss the mechanics, the actual muscular contractive movements that will occur. As you see here in this particular slide, it's an overview of the neuromuscular junction. Synaptic end bulb comes down, synaptic vesicles filled with acetylcholine will be secreted. At the electrical stimulation of a nervous system and this nervous tissue, there will be a release of chemicals across the synaptic cleft into the sarcolemma of a muscle. Sarcolemma and the sarcomere unit is what I'll be describing as far as the actual mechanics, the movement of thick and thin muscle fragments. The filaments, it's important to understand filaments of particular muscle fibers are in contact via the synaptic cleft with the nervous system. We see a motor nerve myelinated coming down, the motor end plates. Some of the basic knowledge and background information that you need in order to understand this concept is that a motor neuron, the muscle fibers that it innervates, so the innervation of the nervous system allows for the contraction of the muscle, the movement of the body. Very important to understand the process involved in the interaction of these two systems. The fine motor control, for example, would be eye muscles. There are 20 muscle fibers per nerve. There's also a gross strength control, and a good example is the gastrocnemius muscle, where there's 1,000 fibers. So the overall understanding of what's going on, the concept of both systems working together to make sure that muscle contraction does take place. There are certain characteristics that are important for the basic understanding of muscle control. Responsiveness is the excitability of a muscle fiber. It has the ability to take the electrical and the chemical stimulation, responding to it, such as acetylcholine, which I'll mention later. The muscle fibers are conductive. The electrical charge will stimulate or excite that particular muscle. The muscles will contract or shorten. The extensibility is the ability of the muscles to actually be stretched, and then a fifth universal characteristic of muscle, the elasticity, where they're able to actually come back to their regular resting situation. Regulatory and contractile proteins. This is the mechanics, the way myosin and actin contract together in unison in a concert type of situation. They will do the work of shortening the muscle. There'll be other muscles or other proteins that are involved in actually controlling, starting or stopping this particular muscle contraction. These regulatory proteins, tropomyosin and troponin, I will explain those and show them diagrammatically as well. Energy is needed. I will mention that the ATP and the breakdown of ATP to ADP plus a phosphate is important for the muscles to contract and for the muscles to relax. Diagrammatically, here's the thick and thin filaments. The myosin with the thick filament is the term. Actin is the thin filament. Once again, basic understanding. And the relationship is what I'm trying to give you an idea of right here. The actual heads of this thick filament, the thin filaments, interspersed between them in a very rigid, comfortable type of fashion. It's important to know that this is a structure which, unless stimulated, does have a competency where it will be able to be stimulated again. There are certain components. I mentioned the actin, the myosin, the troponin, the tropomyosin. There is also connectin, which keeps the thick and thin filaments aligned so that there is a structure to this muscle. It's very important that these thick and thin filaments do stay aligned. The connectin is listed right here and goes down the core of the thick filaments. Thin filaments interspersed, and then a zone where there is only the thick filaments. So we have the overlapping thick and thin, and we have just the thick filaments in the center, all separated by Z discs or Z bands. This is a sarcomere unit, that's what it's called. So we have some basic information. Now really the physiology involved with electrically excitable cells. The excitable cells is the whole idea. Nervous system, copacetic unless there's a resting membrane potential unless it's stimulated. Muscle system, muscle type of cells, resting membrane unless stimulated. It's all due to some ionization and a polarization. Very important for the understanding of a polarization. Positive, negative ions, chemical stimulation. What we're going to be able to take a look at is that the plasma membranes of the sarcomere unit, the actual sarcolemma, will receive acetylcholine. They will have an opportunity to have potassium and sodium rush, with the sodium rushing into the cell, the potassium and other anions removed from the cell. This is that action potential, the excitation of a muscle in order for it to contract and move the body. Some of the physiological background was explained right there with the biochemical interaction of ions. We also have 4 phases, which I'm going to show diagrammatically. 4 phases involved in the process of an actual muscle contraction, the mechanics of how this occurs. A muscle cell, excitation takes place. The action potentials in the nerve lead to formation of action potentials in the muscle. Without the nervous system, like I said, the integration of 2 body systems, providing a homeostatic balance to these systems and the body, that enables muscles to contract. With the 4 phases, we have the excitation-contraction coupling. I'll show a few slides diagrammatically for that. The contraction phase, the actual shortening of the muscle, the movement, and then relaxation needs to take place so that we could possibly have another contraction almost simultaneously. We'll talk about those particular instances where a muscle needs to contract either isotonically or isometrically. Next few images we'll try and explain in certain steps. Essentially voltage gated. I've discussed sodium and potassium, 2 very integral ions in the body, always available, always necessary for the actual body function. Calcium also comes into play. Voltage gated channels where calcium infusion will take place in order to propagate this action potential. I described this here. I go back to the original diagram that I had on the first slide. We've got an opportunity for you to take a look at how the nerve meets the muscle, the interaction of sodium, calcium, and potassium. The sodium gates opening, the potassium being removed from the sarcolemma, right here at this particular plasma membrane, all taking place and allowing for us to understand how the physiology works with the chemistry. Now, sodium, potassium coming in and out of the sarcolemma, all instigated by that electrical stimulation. There will be an infusion of calcium through the T-tubules, which are transverse columns in this particular muscle cell. And the actual muscle fibers will contract along these right here are the key, the myosin and actin. The opportunity for ATP to come into the active sites, myosin hooking on, latching, it's a power stroke is what it's described as, in order to move the thin fibers of actin. You all understand with the ATP necessary, we are going through so that you understand the ATP is necessary to not only ratchet down for myosin and actin, but also to remove and allow the relaxation of the muscle. I'm going to move through for relaxation. It does take place. Acetylcholinesterase takes the acetylcholine away. We have calsequestrin where the calcium is restored. It's important to have this just in case there's a subsequent contraction. Very important for a homeostatic balance with muscles. The mechanics, in the end, essentially 4 phases: contraction, relaxation, and the refractory period for a subsequent important physiological response. Thank you very much.